part of the down payment is that there's a little
I'm sure they use like different sets of there versus other
that we
parts.
can keep it in the same.
There's a little bit of wire. I think rice is the staple that we don't always do. Except unless you have spaghetti, you don't have rice.
I feel like rice is like the staple.
I think French.
So she's buying her own place, so that means she's not tax deduction anymore?
Oh, she's a tax deduction.
You guys.
Okay.
Yeah.
Oh, really?
I don't have any dependents.
Really? The one in medical school?
Maybe back in the game, there was regions, some ideas that made it better, and brought it back, and
Yeah, they just
kind of got heritage or something.
Really?
Yeah.
or something like vice versa or something. And then like, well,
Some of them were still in school, and they won't have a job.
No, I think
I
it
did
ends in a certain
poetry
point.
and my ancestry is all over the world. Like, I was just amazed by it. Like, okay, I'm everything that happened.
It's certainly
Yeah, I don't know what happened there in the history and how, like, politics kind of screws around.
Well, I think with colonization, it's not that hard to get some kind of
Yes.
right? Like someone came in, so what language are you using?
But it's like
It's
all
all
over
about
the
their
place.
size.
Hi.
Hi. My friend, he said that I wrote to yes, we're going to teach Japanese. So it was not like affiliated with genetics.
No.
Oh,
Have you met Philip Gilmar?
I don't know.
Philip works with Larry David, who runs the Proteomics course.
Okay. Especially when places are
You have Japanese?
Yes, I have everything. Even,
Now I want to do mine.
I hope she was coming, but I don't know.
the Native
Back
Americans.
either way.
Okay, well,
Yeah, from the ancestors that cross.
do you know Shannon's
Okay.
Yeah, you get your own
We were till
data,
2:30,
but
so
they don't
okay.
provide-
It's 2:32.
Which one are we finding out that?
More than good.
I don't know. I think there's. I know they don't use like.
I'm sure that the more that you
Gives
do
you
it,
more
the
depth.
The better they get at it. It's not that awful though. Like.
yeah.
I don't know, here is your ancestors.
Wow. But
Or
like,
they would take it and they put it in the case.
no, like.
Oh, there's slaves the whole time. I think for this case,
I know, right?
I have no idea, but there's definitely an industry going on there.
You're from Tehran, right?
Yeah.
Yeah, so we could have like all sorts of like passage in that area at the time, right? So
Microbiome.
But it was like all the way back in the history of the Mongolians and Shanghais, you
it's kind of actually somewhat based, but Iranians consider themselves a big yeah, and they consider themselves like
Yeah, the beginning of.
Well, they-
What you mean
They
French
like
or,
a Canadian race.
it's not true.
It's like Jesus is like around here.
Okay. Okay, let's do it. Okay, we'll go ahead and get started. Welcome, everyone. As often happens at the end of the term, we have a flurry of proposal defenses and defenses. And so today we have a proposal defense from Ravi Madhira, who is a student in the BCB track in the master's program, and his advisory committee over here, Dr. Cohen, Dr. Barrett, and Dr. Phil Walmarth. And Ravi received a honors baccalaureate degree in biochemistry and biophysics from Oregon State. He had a few jobs around the campus and primate center, doing bench research in molecular biology. Then he came over to the dry side and joined our program, and he's in the BCB track. He's also an avid sports fan and enjoys playing cricket. Maybe he'll teach us someday. But what he'll do today is give us his the proposal, and we will question him. This isn't like the formal defense where the audience has to leave and so forth, although his committee may want to ask him questions afterwards. So.
Thank you, everyone, for coming. Today, can everyone hear me? All right. All right. Today, I will be presenting my thesis proposal on improving protein identification and quantification in shotgun proteomics by clustering. So let me begin with defining proteome. The term proteome is coined in the 1990s to mimic the term genome, which represents an entire collection of genes in a system. So a proteome is a set of proteins expressed or modified by cell lines, tissues, organisms, or any biological system. And similar to genomics, then proteomics is traditionally defined as the large-scale characterization of the entire proteome. However, with the improvements in the proteomics field, a more inclusive definition also exists, which is to understand the biological functions such as protein expression levels, post-translational modifications and regulation of proteins, protein-protein interactions, and characterization of protein complexes. Beginning in the 1990s, mass spectrometry has become the predominant tool for protein identification and characterization. And when mass spec was originally restricted for small or thermostable compounds because of lack of ionization techniques that can ionize molecules from a condensed phase to a gas phase. But with the addition of MALDI and ESI, electron spray ionization, mass spec analysis was accessible for large polypeptides. And these ionization methods, in conjunction with high-performance liquid chromatography, or HPLC, allowed highly automated studies on biological samples. And there were also improvements in mass analyzers, such as time of flight, quadrupole time of flight, ion trap, orbit trap instruments that drastically improved the sensitivity and the accuracy of protein and peptide detection. And these instruments now allow us to obtain peptide sequences, peptide abundances, and also post-translational states. And samples with thousands of proteins can be now routinely processed. And finally, like all large-scale, high-throughput methodologies, there are improvements in computational tools such as bioanalytics and bioinformatics that have gone hand in hand with platform development. And so these advances have allowed proteomics to move beyond protein identification and quantification and to a more advanced studies of functional and structural proteomics in complex biological systems. There are two processing methods in proteomics. You have the top-down processing, top-down proteomics, and bottom-up proteomics. In top-down proteomics, a protein, usually an intact protein of about 200 kilodaltons, is characterized using liquid chromatography or mass spectrometry or tandem mass spectrometry. And in top-down processing, it usually requires an extensive protein purifications to get good characterization of the proteins. However, the most widely used processing method is the bottom-up proteomics. This is where proteins are first digested into peptides using enzymes such as trypsin or chymotrypsin, and then the peptide mixture is then subjected to liquid chromatography or mass spec for peptide characterization. And when bottom-up analysis is performed on a mixture of proteins, it is called shotgun proteomics, and the idea is very similar to the idea of shotgun sequencing of DNA. So let me walk you through a typical shotgun proteomics experiment. The experiment can be split into 3 steps. The first step is sample preparation and peptide separation. So in the first step, a sample is first digested into peptides, like I said, using enzymes like trypsin or chymotrypsin. And optionally, these peptide mixtures can be processed to capture a specific type of, specific class of peptides, like phosphorylated or glycosylated peptides. And then these peptides are then separated using the liquid chromatography system. The second step involves peptide sequencing with mass spectrometry. And these peptides, as they elute from liquid chromatography, are then ionized using electron spray, and they're transferred into a gaseous phase. And then the selected ions are then subjected to mass spectrometry. And the result of mass spectrometry is the production of these fragmented ion spectra that can be associated with peptide sequences. And the final step is peptide identification. This is where the spectra that's obtained from the tandem mass spectrometry is compared to a theoretical spectrum that is predicted from in silico digestion of protein sequence databases. So let's take a closer look into step 3 now. A protein search database that usually contains a large number of proteins, such as NCBI or UniProt, it undergoes an in silico enzymatic digestion to generate all possible peptides in the sample. And based on the measured mass of the peptide ion, you can obtain fragmentation models of similar theoretical peptides. And these are the theoretical spectrum. And then on the other hand, what happens is the acquired spectrum, it's first filtered for noise and then normalized before being scored against the theoretical spectrum using search engines such as Sequest or Mascot. And these search engines, they use a proprietary scoring function to rank the peptide to spectral matches, or PSMs. And as you can see from this figure, they've identified 5 candidate peptides that can match to this acquired spectrum. And there's a score given next to each peptide. And generally, the highest scoring peptide is identified as the peptide match for the spectrum. So when proteins are chopped into smaller pieces by trypsin or other enzymes, there can be a loss of connectivity between proteins and peptides. And despite all the advancements in mass spec instrumentation, there is still a difficulty in inferring proteins from the observed peptides. And the digested peptides can fall into 2 categories. You can have unique peptides, which provide immediate information about their parent proteins. So for an example, in this figure, you have peptides 4 and 5, which are only linked to protein 4. So they only have information about protein 4. So from this example, if you see peptides 4 and 5, you immediately know that protein 4 exists in the sample. And on the other hand, you can also have shared or degenerate peptides. These peptides can be linked to multiple proteins, so they provide this confounding protein information. So for instance, in this figure again, if you see peptides 1 and 2, they can be present in both proteins 1 and 2. So you don't know if protein 1 or 2 or both proteins exist in the sample. So this sort of challenge in proteomics is very similar to that in next generation sequencing, where short DNA reads have to be mapped to their originating genes. So the last part of shotgun proteomics is the protein inference step, and this is where you want to identify all the proteins from the observed peptides. And unlike DNA, you cannot really amplify proteins, and also despite the reduced information content that you get from shared peptides, you can't ignore them because you could lose some potential information. So the goal of the protein inference step is to generate a minimal list of proteins that represents an entire collection of the peptides that are detected. Here what I have is 2 common protein inference situations. These are relatively straightforward, but there are several other situations that can cause differences in how proteins can be assigned based on the observed peptides. And in the top case here, we see 3 unique peptides, peptides 1, 4, and 5. And from just this information here, we know that protein A and protein B exist in the sample, because protein A can be defined by peptides 1, 2, and 3, and protein B is defined from 2, 3, 4, and 5. And these are called distinguishable proteins because they are because they're identified by non-identical peptide sets. However, you have a second scenario where you have peptides 6, 7, 8, 9, and they can be matched to both protein C and D. So it's hard to tell if protein C or protein D or both exist within the sample. So these are reported as an indistinguishable group. They're inferred as an indistinguishable group because you don't know if, again, like I said, if protein C or protein D or both proteins exist. And if we had ignored shared peptides, we could have lost information about protein C and protein D. So why does protein degeneracy occur in shotgun proteomics? The first major source of peptide degeneracy is the underlying genomic structure of organisms. Higher eukaryotic organisms have a more complicated genomes, and they have modifications like RNA editing, alternate splicing, and post-translation modifications that can yield similar protein products. So because of these similar protein products, a peptide can be matched to several proteins. And the second source of peptide degeneracy is the choice of the protein search databases that are used in peptide identification. And I've showed you in a couple of slides back how how protein search databases are actually used for peptide identification. So, and there are several protein search databases like NCBI, UniProt, Ensemble, that greatly differ in number of protein counts. So that could lead to differences in peptide identifications and could possibly lead to peptide degeneracy. So the objective of my proposal is that high levels of degenerate peptides may confound basic protein inference logic and also may cause complications in when you're quantifying proteins. One strategy that is used in quantifying proteins is to discard all the shared peptides and use only unique peptides. However, like I showed you earlier, that could cause loss of information. And so what I am proposing here is an implementation and evaluation of clustering algorithm that groups proteins with unique peptide evidence but large shared peptide evidence to improve protein identification and quantification. And in the process, I will contrast protein clustering to standard protein inference to understand the effects of protein search databases and the genomic complexity of the organisms. I'd also like to understand if there is any kind of a biological relevance of the members of the protein clusters that are generated by this algorithm. So before I move on to my specific aims, I'd like to reiterate some definitions that I'll be using throughout my presentation. You have distinguishable proteins, like I mentioned earlier. They are proteins that are represented by sets of non-identical peptides, and indistinguishable proteins are protein groups that are represented by sets of identical peptides. And shared peptides are peptides that can be linked to multiple proteins, while unique peptides are only specific to proteins or protein groups, to one protein or protein group. So the first aim 1A is to analyze protein search databases for protein and peptide degeneracy. As I mentioned earlier, there are several sources of protein search databases that are commonly used in proteomic studies. You have UniProt or IPI that are used for higher eukaryotic organisms such as humans and yeast. And NCBI has a highly curated database with a wide range of organisms. And then Ensemble is also used as a reference genome in transcriptomics, but they also have protein versions. So what I plan to do is to compare protein and peptide characteristics across these multiple protein sequence databases such as UniProt, NCBI, Ensemble, and IPI. And I would also want to contrast protein and peptide degeneracies between higher eukaryotic organisms such as humans and mice and lower eukaryotic organisms such as yeast. And I have some preliminary results from an analysis of 4 protein search databases for human and yeast. And as you can see in this figure here, this is the human protein counts across 4 different databases. There is definitely differences across the 4 different databases. You have UniProt that has about 133,000 human protein counts, while NCBI only has about half of it, 73,000. And in addition, we've also done some theoretical trypsin digestion of all the proteins and computed the shared peptide statuses. And the red boxes that you see here for a human, these are the shared peptide characteristics in all the proteins. And as you can see, across databases, there's about at least on average, 35% shared peptides. So that sort of tells us the complexity of the genome in higher eukaryotic organisms or humans. And this sort of shared peptides is because of the presence of alternate splicing or splice variants. And in contrast, when you look at lower eukaryotic organisms such as yeast, they have relatively smaller protein counts compared to humans. You only have about 6,000 compared to 130,000 here. But there still appears to be some variation in the protein counts across protein databases, with NCBI having only about 5,800 compared to UniProt, which has about 6,500. But the interesting one is the shared peptide percentages. They are very small, only about 2% on average across databases compared to humans, which is about 35%. So this sort of tells us that there is a big genomic complexity difference between humans and yeast. Aim 1B is to analyze proteomic search results from different protein databases for real biological samples. Actual biological samples, particularly from higher eukaryotic organisms, usually represent only a small subset of all possible proteins. And there could also be some processing biases that would favor soluble proteins and soluble peptides, and there could also be some proteins that are very low to detect. So the protein and peptide degeneracies in the sets of proteins detected in biological samples may not really mirror the degeneracy that we observe in the whole protein databases. So what I plan to use diverse data sets of published studies of human plasma cells, cultured mouse cells, and yeast whole cell lysates, and the same protein databases, search databases that I used in AIM 1A to generate identified proteins and peptides within these samples. And I'll use similar metrics that I used in AIM 1A, such as distinguishable and indistinguishable protein counts, and the shared and unique peptides to see if actual samples actually accentuate or mitigate the complexities in the underlying protein search databases. AIM 2 is the heart and soul of this project. The first part of AIM 2 is to implement the proteomic analysis workflow, or the PAW clustering algorithm, to results in AIM 1B. And the protein inference step produces a protein summary report and a peptide summary report. The protein summary report lists all the proteins that are identified within a sample, and there is also additional information for each protein, such as the spectral counts for each protein, and also all the proteins that share at least a peptide with that protein. And similarly, with the peptide summary report, you generate all the peptides that you've identified in the sample and all the spectral counts for each peptide, and also any proteins within the sample that share that peptide. And with the protein and peptide summary reports, the PAW clustering algorithm is then implemented, which performs a pairwise comparison of the sets of proteins that share at least a peptide to test for clustering. And based on shared and unique peptide evidence, proteins may be collapsed into clusters. So there are 3 ways of clustering. This is a pseudo-redundant cluster. In pseudo-redundant clustering, if 2 proteins have a very small unique peptide evidence, but they share a significantly large shared peptide evidence, then these proteins can be clustered together as a pseudo-redundant clusters. And the logic behind that is, if the shared peptide evidence had not existed, then these 2 proteins would have been completely indistinguishable proteins. The second way of clustering is looking for pseudo-subset clusters. In pseudo-subset clusters, if one protein has a very small unique peptide evidence, but the second protein has significantly large unique peptide evidence, but they both share a significantly large shared peptide evidence, then the first protein would be a pseudo-subset of the second protein. And again, the logic behind that is if the first protein did not have this unique peptide evidence, then the first protein would have been a complete subset of the second protein, and only the second protein would have been reported in the protein inference report. And finally, the shared clustering by shared spectra clusters. And this is very similar to pseudo-redundant clusters. And this is a method to identify slightly distant pair of proteins that can still be collapsed into a cluster. And whenever a pair of proteins are clustered, they're combined into a single entry, and the unique and shared peptides are recomputed. And this clustering iteration continues till a stable number of proteins are obtained. So we will evaluate the effects of clustering by processing the results of AIM-1B with the PAW clustering algorithm that I just mentioned. And throughout this process, the sample that we're using is exactly the same, and the sample has a definite number of proteins. But what's changing is the protein search database that we're choosing. And the number of identified proteins must not be dependent on the protein search database. So what we wanted to see if the clustering algorithm generates these proteins and peptide characteristics that are more independent of protein search databases. And so for each protein search database, we will obtain similar metrics as in AIM-1B, such as the distinguishable and indistinguishable protein counts, and shared and unique peptide counts. So let me show you an example of how PAW clustering works. Let's say we've identified 3 proteins, protein A, B, and C, and their respective peptides after the protein inference step. The black-colored peptides that you see are the unique peptides, so they are only specific to that protein. So for instance, protein A has 4 black peptides, and protein B has 1 black peptide, and protein C has 1 black peptide. And the red-colored peptide that you see is common among all 3 proteins. And the blue-colored peptides are only shared between protein B and protein C. So that's how, let's say, we've identified these proteins. And so what we're going to do is implement a PAW clustering algorithm. So like I said earlier, the PAW clustering algorithm performs a pairwise testing to see if a clustering can occur. So it's going to compare protein A and protein B, protein A and protein C, and protein B and protein C. And after the clustering algorithm, what we notice is that protein A is unchanged, but protein B and protein C are clustered together as a protein BC family because they have a significant shared peptide evidence, all the blue-colored peptides, but they only have a very unique or a very small number of unique peptides. And once these 2 proteins are clustered together, the unique and shared peptides are recomputed for this cluster. And when you recompute the unique peptides, what you see is that the unique proteins that you've seen in protein B and protein C still exist, but the 8 shared peptides, the ones in the blue, are now considered unique with respect to the BC family. So that's how the unique and shared peptide statuses change. Or sorry, the unique peptide statuses change upon clustering. So the PAW clustering algorithm is in a way a very straightforward extension of the protein inference process. We also wanted to look at clustering algorithms that are in the literature and compare the PAW clustering with the one in the literature, but to our surprise, we only found 2 other clustering algorithms. One was by the makers of, one was an actual publication by the makers of Mascot. Another one was just a software technical manual by Scaffold. And Mascot uses the unique peptide ion scores and uses that as a distance metric for hierarchical clustering. But Scaffold uses shared peptide evidence between 2 proteins to cluster. So what we thought was actually to implement a similar version of Scaffold and implement that into our PAW clustering framework. So we could use the same protein and peptide summary reports. And another reason would be that if there's any differences that we see between the two clustering algorithms, the scaffold and the PAW, it would only be because of the clustering algorithm and not some upstream processing pipeline. So the scaffold algorithm also performs a pairwise comparison of proteins and clusters proteins based on shared peptide evidence. So two proteins are clustered. In scaffold, two proteins are clustered if they share at least 50% of their peptides. It's a very simple, straightforward clustering algorithm compared to PAW. And some interesting things that we can do is look at or compare the PAW and scaffold clustering results by looking at the peptide and protein characteristics between the two clustering algorithms. We could also look at the number of clusters that are generated by the two algorithms, and also some, maybe differences in the cluster sizes between the two clustering algorithms. And some other interesting things to look at would be see if there are any common clusters that are generated by the two clustering algorithms, or any unique ones that are specific to a single clustering algorithm. So now that I have established a protein clustering algorithm, in this aim, I wanted to see if there is any kind of a biological relevance to the clusters that are generated from this algorithm. And what we wanted to do is to actually perform some internal and external evaluation of the clusters that are generated from this algorithm. So for the internal evaluation, I wanted to calculate the sequence similarity score for each pair of proteins that are in the cluster. And the sequence similarity score will be used as a distance metric for internal evaluation metrics, such as silhouette width. And what silhouette width measures is how similar an object, or in this case a protein, is to its own cluster compared to other clusters. So what I can do is I can calculate the average silhouette width score for all the proteins in a cluster to determine how tightly the clusters are grouped. And I will also research I'm also planning on looking at other cluster metrics, such as maximum cluster distance and the minimum cluster distance, using sequence similarity score again between a pair of proteins to see which can be applied to this problem. And also see if the results actually differ significantly between the PAW clustering algorithm and the scaffold clustering algorithm. So for truly evaluating an algorithm, we need an external method of evaluation, and unfortunately in our situation we don't have a gold standard for evaluating our clustering algorithm. So what we plan to use is some biological information about the cluster members to give some kind of sense of how well the clustering algorithm performs. So in addition to the sequence similarity scores, we also wanted to look at some external information regarding the proteins, such as the gene ontology terms, to see if our clustering has some kind of a biological meaning to it. So we wanted to use molecular function and biological process ontologies to identify any significantly shared geoterms for the protein members in a cluster. And usually the geoterms provide you with a p-value for clusters based on the background set. Based on the occurrence of common geoterms on the background distribution. And for the background distribution, for the background set of proteins, what we wanted to do is to actually use the proteins that are obtained, all the proteins that are obtained from the protein inference step. So the last aim is to understand the effects of clustering and also the protein search databases on quantitative information. And for this, I plan to use TMT labeled mouse samples. The TMT is tandem mass tag, and obtain quantitative information on all unique peptide spectral matches using the Proteome Discover software. And the Proteome Discover software provides you with intensities and the quantitative data. And from this quantitative information, I can compute the quantifiable information content, or QIC, which is just the ratio of unique PSMs out of the total number of identified PSMs. PSM is, again, like I said earlier, it's a peptide to spectral match. And what we wanted to see is how the quantitative results change upon clustering and also across multiple protein databases. And we wanted to see if there are any differences in QIC across protein databases and also before and after clustering. So what we expect is that there will be a greatest change in QIC for highly abundant homologous protein families, such as actins or tubulins or immunoglobulins or keratins, which would have a large number of shared peptide evidence, but a very small number of unique peptide evidence. So when you apply protein clustering to those samples, they would form a family, and all the shared peptide evidence would then be unique peptide evidence with respect to those families. And by doing that, it actually increases the quantifiable information, which is just the count of unique peptides, the counts that you get from unique peptides. So let me walk you through, again, an example of how QIC could be computed. And I'll use the same example that I've showed you previously. And just to recall, the black peptides are the unique peptides, and the red ones are common among all three proteins, and the blue ones are just shared between protein B and protein C. And when there is no protein clustering, QIC is computed right after protein inference. And QIC, like I said, is the total unique PSMs over the total identified PSMs. And in this case, the total number of unique PSMs are just the peptide or unique peptide counts is 6. You have 4 from protein A, 1 from protein B, and 1 from protein C. And the total identified PSMs is 15, which is the count of all the peptides. So the QIC is 6 over 15. Now, when you implement protein clustering, like I showed you earlier, protein clustering is implemented, and you've come up with protein A and a protein BC family. And now the total unique PSMs is still 6, but then these 8 peptides, the shared peptides, are now unique with respect to the family. So then they get added to the unique PSM counts. You get 14, and the total identified PSMs is still 15. So the ratio is 14 out of 15. So this is a simple example where we show how protein clustering actually improves quantitative information. In conclusion, today I have proposed implementing and testing protein clustering algorithms that combine proteins with significant shared peptide evidence and very small unique peptide evidence. And in the process, I plan on investigating the effects of protein search databases and also investigate the effects of genomic complexity between higher eukaryotic organisms and lower eukaryotic organisms. I will compare the PAW clustering algorithm with the scaffold and look at some internal and external evaluation metrics. And also, finally, I would want to see if there compare any effects of protein clustering on quantitative results. So look at any QIC differences before and after clustering, and also across databases. And finally, I'd like to thank Dr. Aaron Cohen, Dr. Phil Wilmarth, and Dr. Uri Barat for their support and their guidance on this project. And additionally, I'd like to thank Dr. Shannon McWeeny, Diane, Dr. Lynn Schwab, and all my BCB classmates and professors. Thank you. And any questions? Yes. No, so the protein counts, we got these from the non-redundant databases. These are straight from the protein search databases, they're direct counts. But the peptides, where we've performed this Pythonic way of trypsin digestion, where you just cleave the sequences between arginine and lysine, K and R. And we just looked at the shared peptide counts, or sorry, the peptide counts.
But those can differ because you're looking at different sets of proteins, right? If you were looking at the same set of proteins, would the peptides be the same? These are proteins straight from the databases. So
I guess they could be different, but I guess the point of this, what I was planning on showing here is the differences in the higher eukaryotic organism compared to a lower eukaryotic organism. And because we're using straight up results from protein search database, I haven't actually thought about if you need to compare similar proteins. This is because in this aim, what we're trying to show is, how protein search databases differ. And
I think the point I was trying to get at is whether one database is better than others, or whether you can combine information.
That would be a hard question. It depends on what type of research that you're shooting for. If you wanted to get all the information about your sample, then perhaps using a larger database would be good. But again, the caveat is that larger databases could have too much information where the shared peptide evidence would be abnormal, and for those kind of situations, you'd probably want to use this kind of algorithm. So, I think by the end of the project, perhaps I'd have an idea on the better protein search database. But for now, I wouldn't know if, or I wouldn't want to say if I'm actually preaching for a specific protein search database. Okay. Yeah.
So I think I'll follow up on the same kind of question. So in the yeast, you said you're seeing almost 6,000 proteins, right?
Mm-hmm.
Are those 6,000 represented in the human? And if not, like how can we compare the two? It's kind of playing on the same theme, but like the problem would be that there are these proteins that maybe, like say hemoglobin, that's multi-compartment. Is that each compartment is in there, or are they an independent protein? And how can you clean that out and filter so you're not doing a lot of duplicate across the species?
So these are called non-redundant databases. What they mean is there's only a single entry of the entire sequence. So all these 6,665 that you see for UniProt, they're all different sequences. And we've actually, I'm not showing you another little part where we've actually looked at. We've actually tested to see if the non-redundant databases are actually non-redundant, meaning that they don't have any duplicate sequences. What we did notice was that there were about, there was a small percentage of proteins that were or sequences that are actually duplicated. But to get back to your question, like I said, these are non-redundant databases. So there's only one copy or one sequence for each protein. And if in their database they show a certain protein having two different sequences, then that would be represented in these graphs also. But usually, if there are two proteins there, I'm assuming they would be two different sequences. They wouldn't have the same exact sequence of two different proteins in the database.
I guess my point was that is the same protein, let's say yeast probably have some alcohol dehydrogenase, right?
Sure.
And I have that too. Is my protein, and I know there's some sequence differences, is represented to some degree in the yeast or the vice versa? Or is there should we do some sort of filtering, say these are the proteins that are common between the species, which will be more indicative of similar comparisons, and these other ones maybe have their own different mechanism or some sort of a different approach to the quantification or the direct effects?
Yeah, I wasn't looking at that kind of difference. I was actually looking at straight up protein, all the protein sequences from these non-redundant databases, and doing a straight up count of how these numbers change across databases.
When you're doing these proteomics, if you want to know what's in the sample based on where it came from, you use a large database from that organism. What you're asking is interesting, but it's a different question.
I see.
So when you're trying to distinguish the proteins, the redundancy or the degeneracy is going to be firstly between the peptide degeneracy within an organism across the database for that organism. So there's also complications in degeneracy across organisms and contaminants and things like that, but that's a next order effect. Other questions?
Okay, thanks.
And in your 5 minutes remaining, you're going to explain to us all how to play cricket.
Well, there is a cricket tournament this weekend in Gresham, so you guys are all invited. We have the playoffs this season. So,
I used to play cricket.
you
in
should
Australia.
all right. Well, I always say cricket is a God's gift.
I mean, they're competitive, right?
I still haven't figured out the role. I did play once, but they told me I was playing more